So I think we'll start. Welcome to all to this webinar jointly organized by the Norwegian Brain Council and the Lifebrain project. My name is Isabelle Budin-Ljøsne. I'm a researcher and a work package leader in Lifebrain, and I will chair this webinar and lead you through this hour of talks with good help from my colleague Mary, who will take care of the technicalities of the webinar. Lifebrain is a research consortium which was funded by the European Union. It started in January 2017. We have 13 partners in 8 European countries, and the project is led by the University of Oslo. And basically the project uses large data sets from cohorts and biobanks to understand how environmental, social, occupational, and lifestyle factors influence brain development, cognitive function, and mental health at different stages of life. And now we are in the 4th year of our project, and we decided that we wanted to present our latest results to the larger community. And we will do so in this webinar. But first of all, I would like to introduce you to Anette Storstein. She's the leader of the steering board of the Norwegian Brain Council, with which we are organizing this webinar, and she will say a few words first. Welcome, Anette.
Thank you, Isabelle, and happy to meet all of you. A few words on brain health. What is good brain health, actually? It's the prerequisite for an independent, socially active, and fulfilling life. There is nothing in normal life that is independent of normal brain function, and yet the brain remains the most elusive organ of them all. There is so much more to be learned. No one is guaranteed a healthy brain, actually, far from, because in fact, brain disease will affect 1 in 3 of Europeans at some time during their life. Dementia and stroke are among the major causes of death, and mental disorders such as depression and headache make up for major causes of disability, in particular in young people. So when I was invited to say a few words now, I really want to emphasize another thing. Brain disease comprises neurological diseases and consequences of injury, but also mental disorders. And this artificial division between mental disorders and neurological disorders is on its way to disappear. So I think this is very natural because brain diseases affect people on so many levels: emotional, physical, cognitive, and also the level of comorbidity. The burden of brain disease affects us all by way of the consequences that brain disease has on the national budgets, on political decision-making, and planning for future healthcare. And the burden of brain disease also lies in the huge impact on life perspectives for those who are affected, their education, their professional abilities, the life quality for these people and their families. So how to accomplish good and lifelong brain health is in fact the major global health challenge for the next decades. So brain health, it's about neuroscience, diagnostic and therapeutic approaches and progress, habilitation, rehabilitation, and coping strategies. However, I always find it a bit thought-provoking that prevention of brain disease has so far received less attention than prevention of other large disease groups as cancer and cardiovascular disease. But still we know that good sleep, healthy lifestyle, and mental well-being are immensely important for the optimal brain functioning. And also we know that in the long term, these are factors that help preventing major diseases as stroke and dementia. So I would really like to stress that prevention of brain disease must begin early and be maintained from childhood through adolescence and into adult life. In fact, good brain health is a long life achievement. So the Norwegian Brain Council is very happy to be a co-organizer of this webinar with the Lifebrain project. And I think that the aims that Isabelle outlined of the Lifebrain Consortium is very much in accordance with the brain health as we define it in the Norwegian Brain Council. So I have looked very much forward to this webinar, and I wish you all a fruitful experience, much learning and good discussion. Thank you very much.
Thank you, Anette, and thank you. We are really on the same page when it comes to brain health, and this is good to hear. So before we start, some housekeeping information or webinar keeping information. The webinar is recorded and it will be made available on the websites of the Norwegian Brain Council and of Lifebrain. You are also all put on mute. If you want to ask questions, you can use the Q&A button that you should normally see on the bottom of your page. And we'll try to spend a couple of minutes between each talk to answer questions. And if we manage to keep a timeline, we also have some time at the end for additional questions. So you can write your questions while the talks are ongoing. If there is no time to answer all the questions, it's not a problem because we will post our answers on the Lifebrain website. We also plan to put a summary of the webinar. So we have 4 speakers today, and our first speaker, I'm very glad to introduce, his name is David Bartrés-Faz. He's a professor at the Barcelona Brain Stimulation Lab at the University of Barcelona, and he will talk about loneliness and memory change across Lifebrain European samples. Please go ahead, David.
Thank you very much, Isabelle. Let me try to share my screen. Is it working? Yes? Okay. Do you hear me? Is it okay? So I start. Thank you very much for inviting me. Very glad to be here this afternoon. So I am the PI for the Barcelona Center in the Lifebrain Consortium, and I will be presenting very briefly some very also recent sub-study. So it's quite the preliminary results yet, but we are very interested in studying the impact of loneliness on memory change across the different European samples in Lifebrain. So just let me put a bit of background. What is loneliness? So when we talk about loneliness, it is a subjective state, negative emotion, so a state of psychological distress and suffering associated to the feelings of being left out, lacking company, or feeling alone. This has been called recently a health pandemic. I'm sure now we are familiar with that term. And it affects 30 million of Europeans, 40 million of Americans, and it has very tight links with health. And recent meta-analytic studies said that it rivals all-cause mortality as other major risk factors very well known, such obesity and smoking. If we look at other European surveys conducted before, we can see that among all the things that loneliness could be associated with, including gender, age, socioeconomic status, income, etc., poor health is #1. So loneliness is really particularly associated with poor health, as it is social isolation, which is a related term. Which are the conditions at risk or what loneliness impacts negatively? First of all, mental health conditions like anxiety, depression, suicide, but also cardiovascular and cerebrovascular diseases, obesity. And also maybe there are some studies suggesting that it increases the risk for dementia and cognitive dysfunction, which will be the focus for the talk today. Loneliness may impact negatively health through different non-exclusively, mutually exclusive pathways. For instance, it has a negative physiological impact on immune and neuroendocrine systems, including the stress hormones, hypertension, etc. But then also also affects more a behavioral psychological part, which because lonely people tend to adhere less to healthy behaviors. So in Lifebrain, we were interested to start to see if loneliness measures in our consortium were associated with memory functioning, particularly in those individuals with 60 years or more. I will now present data on mainly today for the Swedish and German cohorts, but we are also investigating in other cohorts of the Lifebrain consortium. For this study, which I said is still ongoing, we had two first main objectives of questions. The first one is, well, we wanted to see, of course, if loneliness would be associated with memory, and we wanted to see how this would be. We thought that it may be that loneliness would affect memory across all ages. So you can see in the schema, the persons, this is a representative schema. Blue lines are low memories of loneliness, low measures of loneliness, red high rates of loneliness. You can see that in the left part, it will be a parallel effect across age, always having lower memory, those lonely individuals. Or it could be that it would accelerate the age-associated memory decline. That was one question, which would be the kind of the shape of the slope, if any, that loneliness will impact memory with advancing age. Then the second one that we had is, if indeed there were memory loneliness associations, would those be stronger for persistent or constant loneliness measures, or would transient modifications or oscillations in memory rates be more closely or more important to be associated with memory? Let's imagine one person that has a given baseline level of loneliness, and then suddenly one of his very close relative dies, and then he or she has a high increase, temporarily increase of loneliness feelings. Would these oscillations in time be more closely related to memory aspects, or would be the baseline persistent loneliness measures? Those were the two questions. So here's our data, but this is data from the Swedish cohort, the Betula data, not yet loneliness, just to show how the association between memory, verbal memory, which is our main outcome, like recalling sentences or recalling words in neuropsychological standardized tests. As you can see, there is a drop or decline expected and very well known in the literature after age 60. This was our target sample. But this slide is also to show you that the results I will present are adjusted by gender and education, because we saw an effect, also expected. Women outperform men in memory tests, and highly educated outperform lower educated. So we adjusted the results for these two variables. So let's talk about the results on loneliness a bit. First of all, these are the plots showing associations between loneliness measures. You can see in the y-axis, the different composite measures of loneliness used by each center, and then the age in the x-axis. And you can see that there is in general this thick black line, which is quite horizontal. This does not a clear shape, it's quite flat. This means that we didn't find any clear associations between increasing age and increasing feelings of loneliness. There seems to be some effect for the Swedish sample, maybe for the German sample, which are the ones where we have more information. See that the first Swedish and German samples, we have several time measures of loneliness. You can see every line here is a subject where we have different time points, measures of loneliness, as we have for memory. But in the Norway and Spanish samples, we have only one time point of loneliness. But in all the samples, there are little associations between loneliness and age. Then let's talk about the memory results. First of all, we want to see this question, the second one actually, if persistent versus transient changes in loneliness across time. Let's just think that for the Swedish and the German samples, we had at least 3 time points measures for each subject for both memory and loneliness at 3 time points spanning different years. So is the baseline loneliness or the oscillations within these 3 time points more related with memory? So you can see the results here. The stand beta numbers are similar to correlations. So they are negative associations between both measures of loneliness and memory. These are of lower magnitude as expected, clearly, than education and gender. But we can see that for the German sample, persistent loneliness is significantly associated with reduced memory, the trajectories of memory, and more so than loneliness oscillations. And the coefficients are also higher in the Swedish sample for persistent loneliness. So persistent loneliness appears to be more closely related to memory. Finally, we looked at the question of if different rates of loneliness, therefore stratifying subjects that you can see in the slide, among those scoring high in the loneliness questionnaires in the first quartile of loneliness scores, you can see in the red lines, or those showing lower rates of loneliness. We wanted to see if these different stages of loneliness would modify the age-associated memory slope that we have seen before. And you can see here that actually it appears to be the case. We can see more in the Swedish than in the German sample, but the shape is similar of the slopes, that loneliness actually may accelerate the rate of memory decline, particularly as age advances and in advanced age. These results are also adjusted by depression scores. So just to finish, we found for now a very slight increase in loneliness with age. Actually, they are not even significant. We found, however, that persistent loneliness, but not transient oscillation, still to have a greater impact on memory and age-associated cognitive decline. And now we will look for moderators for the variables associated, like social isolation, which is closely associated to loneliness, and also a health risk factor or personality. And definitely we will investigate for the underlying brain mechanisms explaining this association between loneliness and memory. And that's all. Thank you very much. I'm happy to take questions. I will just stop sharing.
Thank you, David. We don't have any specific question right now. I might have one though. I've heard in the UK that you can get dance classes on prescription, so that people who are isolated can go out and meet other people. Would you say, based on your results, that this would confirm the usefulness of such prescriptions?
Yes, I mean, this is true. I mean, there are a lot of institutional and governmental campaigns related to combat the impact of loneliness. And in UK, there was this appointed Ministry of Loneliness actually 3 years ago, and they launched a lot of programs to connect, for instance, elder people lonely with young individuals, or as you say, with connecting social bonds with social activities. So yeah, I think that the results we are showing that loneliness has a negative impact of cognitive function, given that social activity, physical activity, social connection with others may be beneficial for cognitive function. Then definitely I would say it supports the idea of people also from the brain health perspective. So lonely people to reconnect socially with others.
And then we have one question. How did you assess transient loneliness? Was it, for example, loss of husband, wife used as a proxy?
Deviations from the mean measure. So we have like 3 time points for each subject. We took the mean of these 3 measures. This may be spanned, every measure may be spanned 2 years or more years depending on the cohort, but we took the mean measure of loneliness of the three measures that we have in these 2 samples, and then we calculated the deviations, the change from this mean that we found, so the dispersion, the change.
Okay, we have a couple of more questions, and I think we'll keep them for the end, okay, because we need to move on to the next speaker. Thank you so much, David.
Thank you.
Our next speaker, and I ask you to turn on your video and sound.
I cannot start my video.
Of Psychiatry,
you have to do
the University of Amsterdam. And she will be presenting associations of depressed mood and lifestyle with brain structure. Go ahead, Julia.
Yes. Sorry. Yes, sorry about that. Okay. Hello, everybody. Thanks for also inviting me for this talk. Yeah, my name is Julia Binnewies, as Isabelle also told you. And I'm a PhD candidate at the Amsterdam UMC in the Netherlands. And I'm going to tell you a bit about our work on associations of depressed mood and lifestyle with brain structure. So just as some quick background, a lot of research has already been done on the association between depression and brain structure. And all of this research has shown that depression is often related with decreases in brain structure. So the depression diagnosis or also more depressive symptoms have been shown to be related to decreases in brain structure or brain volume of certain regions. And the same has actually been found for lifestyle and the brain. So an unhealthy lifestyle, such as more smoking or more alcohol consumption, less physical activity, or very little or very much sleep, and a high BMI, those measures all have also often been associated with decreases in brain structure and of similar regions as also for depression. However, there's also been a lot of research on the association between lifestyle and depression. And as you could also imagine, depressed patients often have a more unhealthy lifestyle. They don't go out as much. They maybe smoke or drink more, also as a coping mechanism for depression. So you can really imagine that these two, lifestyle and depression, are very closely related. So therefore, we were wondering whether the association between depression and brain structure, for example, and between lifestyle and brain structure, whether these are really independent of each other, or whether maybe the association between depression and brain structure might also be partly explained by the unhealthy lifestyle that these patients often have. And also the other way around, whether the unhealthy lifestyle and brain structure association is actually also influenced by depression. And also what not been studied a lot yet is the association of lifestyle, depression, and the change in brain structure. So over time with aging, it is normal that your brain changes and the structure decreases, which is just the normal aging. However, it is not really known yet how lifestyle and depression influence this brain change over time. So first we wanted to look at this in one of the cohorts from Lifebrain, which is the Netherlands study of depression and anxiety, where we follow people with and without depression and or anxiety over up to 9 years. So we have scanned them 3 times on baseline, 2-year follow-up and 9-year follow-up. And in total, we included almost 350 participants, and have 177 of them have data on the 2 years change, and on the 9-year change, we have 84 participants, and they're around the age of 18 till 58 at the baseline. So then for some first results within NESTA, there we actually indeed found what has been described earlier, that depression, and that is depression diagnosis, but also the depression severity of the symptoms, have been related to decreases in brain structure, and then mainly decreases in prefrontal brain regions. And also for lifestyle, we found that, for example, a higher BMI was related to decreases in prefrontal brain structure. But we also found that, for example, a moderate alcohol consumption was actually related to increased brain structure compared to people that didn't drink any alcohol at all. However, for physical activity and sleep duration and smoking behavior, we didn't find any associations with brain structure. And then we wanted to see whether these associations that we did find, whether they were independent of each other. So if somebody with depression and a higher BMI still shows the association of depression and brain structure that we found. And this was indeed the case. We found that people, that depression was still associated to brain structure, and the same was for BMI, even then when we corrected for the other one. So these were independent of each other. And also for moderate alcohol consumption, this was the case. It was also independent of the other lifestyles and depression. Then for the longitudinal analysis, we didn't find any strong associations with brain change over time of lifestyle and depression, unfortunately. However, we did find some small trends of physical activity and BMI and brain change. And here, less physical activity or higher BMI were related to more change over time. So again, in some prefrontal brain regions. However, because we did a lot of analysis and looked at a lot of variables, we needed to take into account that these findings might be due to chance because we did so many analyses. So if we corrected for that, then these associations were not there anymore. So yeah, we did find some trends, but the evidence wasn't very strong, unfortunately. So then for some conclusion. So we found that depression and BMI both seem to impact brain health, and that this is independent of each other. So depression and BMI are both important for brain health. And we did find a positive effect of moderate alcohol consumption on brain health. However, a small side note for this, for the interpretation of this, is that non-drinkers, which we compared the moderate alcohol consumption to, they might not be the healthiest population. So you could imagine that people that don't drink any alcohol at all are not really the general population, but they might have some somatic diseases because of which they don't drink alcohol anymore, or for some other reason they might not drink anymore. So we have to be a bit careful with this interpretation. And then we didn't see any predictive value of depression and lifestyle for brain change over time. So we can see that the associations that we did find, that the causality is still unclear. It might be for depression that the effects of depression happen very, very early on, the effects on the brain, or that maybe even the brain structure that we found to be different or related to depression might not even be related to the disease, but be a risk factor for the disease and not related to the onset of the disease. So therefore, it is really crucial to look more into prevention and early treatment to try to see when the effect on the brain occurs. And of course, we would like to prevent this effect. And then we did find the trends for the physical activity and BMI. So there is something to say that it might be that a healthy lifestyle now does lead to a healthy brain later on, which would be useful for policies, of course. Then we could act on the lifestyle to try to prevent brain diseases, but more research is definitely needed there. And that is also what we want to do within Lifebrain. So we also want to look at the other cohorts that we do have in Lifebrain, where we have a total sample size of 3,300 people. So we want to look there more in depth also at the lifestyle and depressed mood, and also within this the current sample that we did the analysis in the Nesta sample, we have clinically depressed people, but in Lifebrain we also have a lot of participants with subclinical depressed mood. So this might give us a bit of a more generalizable view on how depressed mood in the general population relates to the brain. And also within Lifebrain, we have a much broader age range than we did in the Nesta sample only. So then we also would like to see what the effect of age is on all of these associations. So thank you for your attention. If you have any questions, I can answer one now, or you can also email me or ask in the question and answer part. Thank you.
Thank you very much, Julia, for a very clear presentation. We have one question. What did you measure as brain structure?
I measured, I don't know what your background is, but we use free surfer measures. So that's MRI scans that we use to get the volumes and thicknesses of certain brain regions, and then we used some of those brain regions as outcomes measures. Don't know if that answers your question.
Okay. We have another one. How do you estimate causative relation of structural change with depression? You arose go both ways.
Yes, well, for the analysis where we did find the associations, we don't know which direction they do go. So we can't say anything about the causative relation based on those. So that's why we did the longitudinal analysis as well, but where we tried to predict based on depression at one time point, predict the brain change, but we didn't find any thing there. So yeah, we can't say anything about the causal relationship.
Okay, I see we have one more question, but we'll keep it for the end. Thank you very much, Julia. And we move on to our next speaker, Anders Fjell. He is professor at the Center for Lifespan Changes in Brain and Cognition at the University of Oslo. And he will tell us about how sleep and the brain are related. Go ahead, Anders.
Okay, am I on? Does this work?
Yes, it works fine.
Okay, excellent. So I'm Anders Fjell, and I'm going to tell you a little bit about how sleep and the brain are related. And this is one of the things we focus on in Lifebrain. What is the role of sleep for brain health in the modern society? And there's a worry in society that people are sleeping too little. This is recommendations from the National Sleep Association in the US, where they have tried to find out what are the required numbers of sleep every night to have a healthy body. And they in general recommend quite a lot of sleep to be on the safe side. However, we do not know so much about how much do you sleep and how does the sleep quality impact your brain health? Or is the causality the other way around? Is it so that people with brain health that are not optimal have problems with their sleep? So these are things we are looking at in Lifebrain. So what you know is that prevalent age-related brain disorders such as Alzheimer's disease and Parkinson's disease are associated with changes in sleep patterns. So people with a brain disease usually report worse sleep than other people. However, we do not know what is causing what. Everything that affects the brain in a negative way will probably also affect your sleep pattern in a negative way. But then there are also studies, especially from animal and rodent research, suggesting that if you keep the animals awake, it will have a negative effect on the brain. So both of these explanations may be true, that bad sleep may affect your brain and worse brain health may also affect your sleep patterns. What we also know is that changes in sleep are normal in aging, also in general, not only in diseased or demented aging, but in the aging that happens to all of us. So in Lifebrain, we've asked people, how do they consider their sleep? And for instance, this is answers from several thousand people in Lifebrain. We have age on the x-axis and sleep quality on the y-axis. So a high score is lower quality. What we see is that people in general actually report to have higher sleep quality when they get older. So they have less sleep problems and are more happy with their sleep. At the same time, they also report to feeling less tired during daytime. So young people report to have worse sleep and feeling more tired during daytime. This may mean that older people actually get the sleep that they need, and they don't need to sleep anymore, even though they, on average, sleep for shorter hours. This is an interesting hypothesis. So we're interested in looking at how your sleep habits are related to changes in the brain. Are sleep and brain health related in normal adults at all? So in an older study that we published 6 years ago, we followed about 150 people over time, and we measured how they considered their sleep to be and how their brains changed. What we found was that those who reported worse sleep also showed more brain volume loss over time, and these were normal controls. These were not people with Alzheimer's disease. So in these regions we can see here, which are red and yellow, we found volume loss over 4.5 years in the poor sleepers. However, this relationship was not particularly strong, and we only saw this for adults above 60 years, as shown here with the blue line. Those with more brain volume loss have a higher value, and those with a higher value on sleep, they have more sleep problems. So for people above 60 years, there was a relationship between volume loss in the brain and sleep problems. In Lifebrain, we have focused specifically on a structure called Hippocampus, which is shown in yellow in the figure to the right. You can see this is deep embedded into the brain. And the reason why this is interesting is because Hippocampus is absolutely critical for memory. If your Hippocampus doesn't work, your memory will be almost non-existent. And also Hippocampus is the structure that's the most sensitive to decline in Alzheimer's disease. And we also see a lot of changes in hippocampal structure in normal aging. So that's why this is a very interesting structure to look at in relation to aging and to sleep. And also a lot of the things that are happening while we're asleep is happening within the hippocampus. This is where memories are consolidated or made more stable in the brain. And a lot of this happens while we are sleeping. We looked at this in Lifebrain. And what we can see to the left is a typical change in hippocampal volume across life in normal people. We see that while not much is happening with the hippocampal volume during adult life, it really starts to decline when people are approaching 60, 70, 80 years. And this is a feature we see also in normal aging. We see individual data points to the right. You see that people tend to go down when we follow them over time. There's a lot of individual variations. There are 80-year-olds with hippocampi that are bigger than the average of 20-year-olds. We also have people who really show decline. We were interested in looking at, are there any relationship between people's sleep quality and how hippocampus changes over time? And we found that, indeed, there was a relationship. On the one axis here, we see how much of the volume of hippocampus is lost every year. And we see age on the other axis. And then we have divided people into the good sleepers and the bad sleepers. And what we see is that those who report good sleep, on average, loses less hippocampus every year than those with bad sleep. Again, these relationships are not particularly strong, but they're quite robust in this rather large sample that we have. And in contrast to the slide I showed for the cerebral cortex, which is the outer layer of the brain, see that this relationship is quite stable across life. So the relationship between sleep and hippocampal loss is the same for 20-year-olds, 40-year-olds, 60-year-olds, and 80-year-olds. There's no change over life. We also wanted to look specifically at sleep duration. And in Lifebrain, we did not see that sleep duration had any relationship to hippocampal loss. So we went to another sample that we had access to from the UK Biobank, which has 21,000 participants. And in this sample, people are not followed over time, so it's a one-time point examination. And for most of the questions related to sleep, there were absolutely no relationship with hippocampal volume. The size of hippocampus was not related to how people reported their sleep to be, with one exception, which was the sleep duration question. And here we saw that for most people, those who slept regular hours, there was no relationship between the volume of hippocampus and the sleep duration. But for the real outskirts, those reporting sleeping 4, 3, 4, maybe 5 hours a day, for those who reporting sleeping 10, 11, 12 hours a day, there was a relationship. So that very short and very long sleep duration was related to smaller hippocampal volume. Not volume change, because this was a one-time put examination. And these data show us that within regular sleeping hours, there's no relationship between sleep duration and hippocampal volume. In the outskirts, there was a relationship. Interestingly enough, sleeping very long hours was just as bad as sleeping very short hours. But it's important to stress that this is not a study of causality. We do not know whether sleep actually has an effect or whether this relationship is seen because some people are worse off, and this affects their sleep, and it also affects their hippocampal volume. Sleep is also interesting, as I mentioned, in relation to dementia disorders such as Alzheimer's disease. And Alzheimer's disease is associated with changes in sleep patterns. And again, we do not know what causes what, and probably the causality goes both ways. So bad sleep affects your brain and increases your Alzheimer's risk, possibly, but also having a disease like Alzheimer's disease affects your sleep pattern. We and others have seen that self-reported sleep problems are related to more amyloid in the brain, and amyloid is the main biomarker for Alzheimer's disease. Again, I need to stress that this relationship is not strong. So for an individual, there's no reason to worry about having an increased risk of Alzheimer's disease because your sleep is not as good as you want it to be. Worrying about your sleep will probably affect your sleep negatively, and there's no reason to, on a personal level, do this. Although in a scientific and societal perspective, these relationships are interesting. So what we do know is that sleep seems to have a robust short-term effect on your memory. So what this slide is showing is that after a good night of sleep, people report much better performance. And they show much better performance on the memory test than they do before sleep. So sleep definitely seems to have an effect on strengthening material that is also learned from beforehand. So sleep definitely has a short-term effect. What we're not sure about is the long-term effect of sleep. So to conclude, worse sleep seems to be moderately related to more brain volume loss. For the cerebral cortex, this is the case only in older adults, about 60 years. Sleep duration, which is the number of hours spent sleeping each night, seems to be very weakly related to brain volume. So we conclude that sleep may play a role in brain health. Short sleep duration within reasonable limits does not seem to have a large negative impact on the brain in the long run, but we need a better understanding of the relationship between sleep and brain health across lifespan. So thank you, Anna. May take some questions.
Thank you very much, Anders. We actually have one question. Is the UK biobanks over 21,000 samples hippocampal volume data controlled for age?
Yes, we do control for age in all of our analysis. And we also look at whether age is interacting, so that whether there are relationships that are stronger for older and younger. So yes, we control for age.
Okay, thank you. Thank you very much. Excellent presentation. We're running a little bit late, but we still have one presentation from Christian Drevon. He's professor at the Department of Nutrition, University of Oslo and Vitas. And his presentation is about the use of blood biomarkers for brain health. And we will show you his presentation. It is pre-recorded because he couldn't join today. But you can still ask your questions and we will forward your questions to him. So please, Marie, if you could just start it.
Dear viewers, my name is Christian Drevon. I'm going to talk about biomarkers of brain health. I'm a work package leader in Lifebrain Consortium, and one of the responsibilities is to organize analytical activity based on blood samples collected in the Lifebrain project. Vitas is a contract laboratory analyzing blood samples and other biological samples spun out of University of Oslo. With 26 years of existence, with experience basically on biomarkers in blood and other biological samples. We have been focusing on brain health in this project. And if one look at risk factors or beneficial factors for brain health, the list of 12 points on the left-hand side is pretty much agreed in many leading research institutions. This is a modified list taken out of Harvard Medical School. And you can see that the points include getting and giving stimulation, exercise, diet, reasonable use of drugs, head protection, and having a good sleep pattern. Basically, what is healthy for the heart and body is also healthy for the brain. This is some sort of a guidance for what to look for if we are going to find out what is risk factors we can describe in blood samples for brain health. And the technology Vitas has used in sampling blood in the Lifebrain consortium is based on fingertip blood samples collected on filter papers and dried, the so-called dried blood spots, or DBS for short. It's based on the fact that if you have sensitive methods like we have, you can get by with very small amounts of blood and still analyze many different compounds. So one blood drop contains about 50 microliters, which is 1/20th of 1 milliliter. And you can send this sample in regular mail. You can do many different of methodological approaches like ELISA, mass spectrometry, and chromatography. And you can have the advantage of the consumer or the client, him or herself, taking the samples without any health professional support. This is very beneficial, especially in situations with infectious diseases like we have had recently. The other advantage is that these DBS samples are stable on the special paper we use. More than 200 analytes have been validated on this method. Also, we have developed a method for measuring antibodies against COVID-19. If we look at specific biomarkers that are important, partly already demonstrated in limited studies, and also potential markers for brain health, I have a short list here, including vitamin D, cholesterol, types of fatty acids, especially omega-3 fatty acids, a marker of the mean blood glucose level over long periods of time, called HbA1c, which is glycated hemoglobin. It is a marker of the risk of having high glucose concentration, i.e. diabetes. Then we also measure a large sample of different cytokines, about 13 of them. These cytokines are important, especially when it comes to inflammation, and a few factors are important specifically to the brain as well. One is called brain-derived neurotrophic factor, BDNF. And the other inflammatory markers are especially interleukin-6 and CRP. CRP stands for C-reactive protein and is a general marker of inflammation or bacterial infection in the body. The most recent development in metabolomics, that is generally the science of this biomarker field, is called lipidomics. It includes all lipids you find in a blood sample, and we talk about several thousand different lipids. We can do in the Lifebrain project the screening of these lipids, and we have a special focus, for example, on diacylglycerol, DAG, as you can see here, with a certain number of carbon atoms, namely 36, and 2 double bonds. This lipid fraction is supposedly important for for sleep function. Let me just give a few examples where we have shown that certain dietary factors are important for brain structure and function. The first example is from a study related to prematurely born infants with the body weight at birth or less than 1.5 kilograms. We did a randomized study where we divided the group into different groups, and that is one group got regular supply of nutrients according to general standards of the newborn infant care units, and the other group got an increased supply of amino acids as proteins, energy, more vitamin A, and more essential fatty acids like omega-3 as well as certain omega-6 fatty acids. When we evaluated these infants after intervention with the special nutrients that we think are important for brain development, we used MRI, magnetic resonance imaging, which can give information about the structure of the brain. And to make a long story short, we could show that the intervention group with enhanced supply of essential nutrients got an increase in their development, a more rapid development of certain parts of the white matter, representing connection between different regions of neurons, as you can see on the upper slide. Specifically, it is one pathway that was really standing out. We also examined function in addition to the structural changes. And we used 128 electrodes on the head of the infants, along with the visual set of signals. And we could measure the response to the visual signals by the 128 electrodes. And as you can see on the right-hand side in the bottom of the panel, the intervention group had significantly higher responses to the visual signals compared to the control group. In another study, in collaboration with Walhovd et al., we could show that during a period of, in average, 3 years, with MRI detection, could show that if you have a high concentration of DHA in the blood, you have reduced cortex thinning, which is supposedly a good beneficial factor. And if you compare vitamin D concentration in blood to development of a cortical thickness, you can see that in certain areas of the brain, you also have a beneficial effect of having high vitamin D concentration in the blood. On the other hand, we could show that a high concentration of cholesterol or a high BMI, i.e. a high amount of body fat, will reduce cortical thickness or increase cortical thinning in certain specific areas of the brain, demonstrating that nutrients or lifestyle is important for structural changes during at least a 3-year period. To sum up, many of the present markers of brain health are markers of general health. What is healthy for the body is usually good for the brain as well. In addition, we are searching for more specific markers of the brain, and these could perhaps be diacylglycerol related to sleep and BDNF related to growth and differentiation of neurons. Thanks for your attention, and if you have questions, they may go to the depicted email address. Thank you.
So if you have any questions, don't hesitate to send them to Christian or send them to us in this chat. We will forward them to Christian. We are ready to wrap up. There is one last thing I would like to tell you about, and I will share my screen to show you the website of Lifebrain, the Lifebrain project. We have a study that we are conducting called the Global Brain Health Survey. It is a survey to collect information about how people think about brain health and what they are willing to do for their brain. We've been conducting this study for some time now, and we have collected 17,000 answers so far. And if you are interested in this study, you can take it by just logging or using the link to the website. The study is open to anyone, so please feel free to take it and answer it. It will be open until the end of August. With this, I think we will conclude. There is one last thing that I would like to do, if I just find out how I do this. We have prepared a little poll, and it would be very nice if you could take a couple of minutes to answer it. I hope that you found this webinar interesting. We will put the webinar on our website, so it will be possible to watch it again. And if you have any questions, please do not hesitate to contact us.